So as Mohammed indicated, I will be focusing primarily on preclinical considerations for antibody-based therapeutics. So I'll be providing a brief introduction and then address several themes as part of my talk. One of them would be focus on antibody modes of action, considerations for antibody target selection, different types of antibody platforms and antibody generation, focusing also on xenograft models and considerations for different types of models, and then the last part of my talk, discuss a little bit about some FDA-approved therapeutic antibodies as examples. So I'll be focusing primarily on applications of monoclonal antibodies in cancer, although of course monoclonal antibodies can be used in other disease settings, such as autoimmune disease. Generally, monoclonal antibodies block the interaction between a cell surface receptor and its cognate ligand. In the context of cancer, antibodies can treat the disease in a variety of different ways. One is enhancing a patient's immune response. For example, in an autoimmune disease setting, you might want to actually inhibit or mitigate a patient's immune response, as an example. An antibody can also act directly against cell growth factors that might be present or a tumor cell that might be dependent on such factors. An emerging area within oncology is targeting the tumor vasculature. And of course, antibodies can be used as delivery mechanisms for toxins or radioisotopes. Some of the well-known current monoclonal antibody therapies are currently used to treat a variety of different cancers. Examples here that I've listed are breast cancer, like Herceptin, non-Hodgkin's lymphoma, rituximab is an example of that, chronic lymphocytic leukemia, colorectal cancer, and non-small cell lung cancer. So I've listed here a number of points to consider when one embarks on developing an antibody drug. Some of these considerations include function of the antibody. You want it as an agonist or an antagonist. There are affinity considerations, which Scott Claycamp will highlight in his talk. Epitope selectivity. The isotype of your antibody is an important consideration, which I'll address in a few minutes. Tissue distribution of the target that you're generating your antibody against, as well as species cross-reactivity, which is an important consideration when developing disease models, either in a rodent system or ultimately in a primate system.